let us discuss about digestion and absorption. Let us discuss under the following headings: introduction, mechanism of digestion and absorption, digestion and absorption of carbohydrates, proteins, fats, absorption of water and minerals in the applied aspects. So the basis for the digestion is hydrolysis. The, it is the addition of water to the nutrients. So digestion involves the breakdown or hydrolysis of nutrients to smaller molecules that can be absorbed in the small intestine. So the main aim of the digestion is to make the nutrient absorbable, which happens in the small intestine. So digestion is breaking down of the macromolecules into smaller molecules. So the final product of the carbohydrate is the monosaccharide. Protein is digested into amino acids and small peptides. The fat is digested into fatty acids and monoglycerides. There are different types of digestions. So the first one is luminal or cavital digestion, which happens in the lumen of the gastrointestinal tract, and the enzymes from the salivary gland, stomach, and the pancreas helps in digesting the food. And the another type of digestion is membrane or contact digestion, which happens in the fresh borders of the small intestines. So the enzymes which is present on the fresh border of the enterocytes helps in this. And then intracellular digestion. So that happens with the enzymes called dipeptide peptidases and tripeptidases. There are digestive enzymes which plays an important role. So there is salivary amylase which is secreted in the saliva, pancreatic amylase, and the enzymes secreted in the intestine. These are the enzymes which acts on the carbohydrates. So the intestinal enzymes are sucrase, maltase, lactase, trihalase, and alpha dextrinase. In coming to the protein digestion, so the enzymes present in the stomach is pepsin. There is no enzymes to digest the protein in the saliva. The oral cavity have no protein enzymes. So, and in the pancreas, there is trypsin, chymotrypsin, carboxypeptidase, and elastase. And in the intestine, it has enterokinase, amino oligopeptidase, and dipeptidase. And the enzymes to digest the lipids are lingual lipase, but it has very minimal role to play on the digestion of lipids. The major part of the lipid enzymes are secreted by the pancreas. They are lipase, colipase, phospholipase alpha 2, and cholesterol esterase. And there are intracellular enzymes to digest the proteins. They are dipeptidases and tripeptidases. Coming to the mechanism of absorption, there are 4 mechanisms which are critical for the transport of substances across the intestinal membrane. So the first one is the active transport by means of primary and secondary, and then passive diffusion, and then facilitated diffusion and endocytosis. Endocytosis happens in the infants and not in the adults. So the single most important process in small intestine to make absorption of nutrients possible is the sodium electrochemical gradient across epithelial cell boundary of the lumen. So there are different sodium channels which helps the intestine to absorb the carbohydrates and the proteins. So there is sodium glucose channel, sodium glucose linkage transporter, and sodium amino acid channel. So these are very important channels to carry the nutrients. Sites of absorption. So in the stomach, ethanol and the drugs like non-steroidal anti-inflammatory drugs like aspirin, paracetamol gets absorbed. So in the duodenum and jejunum, so various nutrients, vitamins, various ions, water and electrolytes gets absorbed. So in the ileum, it absorbs the bile salts and the vitamin B12 or cyanocobalamin. So it has major clinical significance. We'll see that later. So colon absorbs mainly the water and the remaining electrolytes. Rectum absorbs the drugs like steroids and salicylates like paracetamol. In anatomical basis for absorption, total surface area of small intestine is 250 to 400 square meter. So we know that small intestine is only 4 meter long. So the folds of Kerfring is 3 times more. And you can see the villi in it, which is 10-fold more. And you can see the microvilli in the from the villi, which is 20-fold more in surface area. So the total of 1000-fold increase in the surface area happens because of these foldings. So which helps to absorb the nutrients better. So the size of a tennis court is only 196 square meter, which is uh, half of what the small intestine is, and lung is only 50 to 100 square meter. Villi, which is a very important structure to absorb the fat. So here you can see the central lacteal, which is very important to absorb the fats. So this is nothing but the lymphatic duct, which absorbs the chylomicrons. And here you can see the countercurrent mechanism which is present in the villi also. So there are pathways for the nutrients to get absorbed. There are 8 barriers for the nutrients to cross it and absorbed by either the blood or the lymph. So there is an unstepped layer, here you can see, and then glycocalyx, and then apical cell membrane, and then cytoplasm of the enterocyte, and then basolateral cell membrane, and intercellular space, and then the basement membrane, finally the wall of the capillaries or the lymphatic vessel. So it has to a nutrient has to cross 8 barriers to reach the blood or the lung. Dietary carbohydrates. So the dietary intake per day is 250 to 850 grams, so which is 50 to 60% of the diet. So polysaccharides, mainly the starch, the carbohydrate reserve of the plants, which is vegetarian source, and glycogen is a non-vegetarian source, which has two linkages. One is to 4 alpha linkages and one is to 6 alpha linkages. And then cellulose is also a polysaccharide, which is a plant source, but this cellulose has no enzymes in the human to digest it. So cellulose get into us, we eat cellulose by plant source, and without digestion, without absorption, it leaves us in the pieces. And then oligosaccharides, they are sucrose, lactose, and maltose, and monosaccharide, glucose, fructose, and pentose. Digestion of carbohydrates. So in the mouth, mainly starch digestion happens. So maltose digestion to starch gets digested into maltose by salivary amylase. So this salivary amylase acts on 1 is to 4 alpha linkages and not 1 is to 6 alpha linkages. Only on 1 is to 4 alpha linkages. So in the stomach there is no enzyme to digest the carbohydrate, but still the salivary amylase continues to act until the pH changes below 4. So we know the pH of the stomach is 1.5 to 2 due to the gastric juice. So the mixing of the food with the gastric juice takes some time. So until then, the salivary amylase can act in the stomach. Once the pH goes below 4 from 6 to 7, so the action of the salivary amylase stops. Digestion of carbohydrates in the small intestine. So the pancreatic amylase is released in the second part of duodenum in alkaline medium. Polysaccharide, starch, and glycogen gets converted into oligosaccharides, that is maltose and dextrin, by the pancreatic amylase. Here you can see, so the salivary amylase does around 20 to 40% of the digestion, whereas the pancreatic amylase does 50 to 80% of the digestion. In the brush border enzymes of the small intestine like dextrinase, maltase, sucrase, and lactase converts the dextrin to glucose, maltase converts maltose to glucose, and sucrase converts sucrose to glucose and fructose. Lactase converts lactose to glucose and galactose. So coming to the absorption of carbohydrates, the site of absorption is from the mucosal surface of jejunum and upper ileum, not duodenum. And the mechanism of absorption is sodium dependent active transport. So glucose and galactose is absorbed by sodium dependent active transport, and fructose gets absorbed by facilitated diffusion, and pentose gets absorbed by simple diffusion. What happens to the glucose? What is the fate of the glucose in our body? So it gets stored as glycogen in liver and muscle, that is around only 5%, and around 50 to 60% is used to produce the ATP. In 30 to 40% is converted into fat and stored in the adipose tissue. Coming to the abnormalities, so lactose intolerance, so the absence of the enzyme lactase, so the lactose will not get digested, so that leads to diarrhea and electrolyte imbalance. So if lactose is not digested, so that will be a thick film which leads to osmosis. So the more fluid gets into the lumen and that causes the diarrhea. So the diarrhea causes the electrolyte imbalance. Secondary lactase deficiency, so that causes intestinal distension, flatulence and diarrhea. So lactose intolerance happens in the infants, which is a bit difficult to diagnose, whereas secondary happens in the adults, which is much easier to treat. Treatment is to avoid the milk products, the dairy products, which has the lactose. Coming to the proteins, the dietary sources of protein. So the exogenous sources are meat, fish, egg, milk and soya beans. The daily requirement is 0.5 to 0.7 grams per kilogram. The endogenous sources comes from various gastrointestinal secretions and also present in desquamated epithelial cells of the digestion of proteins. So in the oral cavity there is no enzyme to digest the protein, whereas in the stomach it has pepsin. So pepsin is a very important enzyme to digest the protein. So 10 to 15% of the digestion happens in the stomach. So the hydrochloric acid converts the pepsinogen to pepsin. So for the pepsin to act, hydrochloric acid's presence is very important. Without that, pepsin will not act. So which breaks down the macromolecule, the protein, into peptones and proteases. And the optimum pH for the pepsin to act is 2. So in the small intestine there are pancreatic proteases, various pancreatic proteases, so which digest the proteins, the peptones into dipeptides, tripeptides and the polypeptides. And the enzymes in the breast border of the intestine like dipeptidases, tripeptidases and the nucleases, they digest further and the amino acid is formed. So there are intracellular peptidases like dipeptidases and tripeptidases which helps in the digestion of protein to form the amino acids. Digestion of nucleic acid and nucleoproteins. So nucleic acid and nucleoproteins present in liver, kidney, pancreas and yeast. In the stomach, hydrochloric acids act on nucleoprotein and forms protein and free nucleic acid. In the small intestine, this free nucleic acid by the pancreatic enzymes like ribonucleases and deoxyribonucleases gets converted into nucleotides and nucleosides. And the enzymes in the breast borders like nucleotidase and nucleosidase, they convert the nucleotides and nucleosides into purines and pyrimidics. Coming to the absorption of proteins, again the sodium dependent active transport mechanism plays a very important role. So the amino acid is transported into the enterocyte by means of the sodium channels. So the transport of amino acid into the blood capillaries from epithelial cells, it happens by simple diffusion and facilitated diffusion. So in the interstitial space, by simple diffusion, finally it reaches the capillaries. Abnormalities of protein digestion and absorption. So inadequate absorption of protein, so that can happen due to the deficiency of the trypsin, and malabsorption can happen due to the deficiency of the sodium transporters. Digestion and absorption of fat. So the dietary fats plays a major role. So daily intake is 25 to 160 grams. So the types of the dietary fats are simple include triglycerides and cholesterol, and compound fats include phospholipids, and associated fats include the steroids. So the main site for the digestion of fat is the small intestine. So the mechanism of digestion of fat has three steps. The first one is the emulsification of fats by the bile salts, hydrolysis of fat droplets by pancreatic and intestinal lipolytic enzymes, and acceleration of fat digestion by visceral formation. Emulsification of fats by bile salts, it is the first step. So emulsification is breaking of large droplets into small droplets by the bile salts, which is must for the pancreatic lipase to act. So it is done by the bile salts. So lecithin greatly enhances this action. So emulsification is breaking the larger fat into smaller one by the bile salts. So bile salts gets attached. So after breaking it gets attached to it. So it so here you can see the large droplet, fat droplet gets into the smaller one, many smaller one. You can see the outer portion which is blue, which is water soluble. They are negatively charged, but they are water soluble or hydrophilic groups. And you can see the inner part. Inner part is lipid soluble or hydrophobic, so which can have cholesterol inside, but outside it is water soluble, inside it is lipid soluble. So outside it covers, here you can see this is the bile salt, this is how it covers, and here you can see this is the lecithin, so which helps in this. So outer layer is easily to remember, outer layer is water soluble and inner layer is lipid soluble, and inside it you can see the small lipid droplets. So the next step is hydrolysis of fat droplets by pancreatic and intestinal lipolytic enzymes. There are three types of pancreatic enzymes. One is pancreatic lipase, which hydrolyzes almost all the triglycerides to two fatty acids and two monoglycerides. Second one is cholesterol ester hydrolase, which converts the cholesterol ester to cholesterol and fatty acid. And then the third one is phospholipase alpha 2, which hydrolyzes the phospholipid and separates the fatty acid. Acceleration of fat digestion by micelle formation. So micelle, they are small water soluble cylindrical disc shaped particles, so composed of fat globule in the center surrounded by 30 molecules of bile salts, so which we have seen earlier. So this is called as a mixture. So the outer layer is the water soluble, inner layer is lipid soluble. Inside it has the fat droplets by means of monoglycerides and fatty acids. So monoglycerides and free fatty acids are incorporated in the central fatty portion. So first fat gets emulsified with the help of bile in the then the pancreatic lipase emulsifies the fat further into fatty acid and to monoglycerides. Coming to the absorption of fat, so the dietary fat which we eat as large either triglyceride droplet or the fat cholesterol droplet, mostly the triglycerides. So they gets emulsified with the help of the bile salt. So these then the pancreatic lipase. So these lipases hydrolyzes the triglycerides into monoglycerides and free fatty acids. So these water insoluble products are carried to the luminal surface of the small intestine epithelial cells. Here you can see. So the water soluble micelles, these are called the micelles, which are formed by the bile salts and other bile constituents. So when the micelle approaches the absorptive epithelium, so the monoglycerides and the free fatty acids, they leave the micelle and passively diffuse through the lipid layer. Here you can see. So here it is water soluble in the lumen, so it easily goes near the brush borders, and from the brush border it is easily absorbed. It easily absorbs the small droplets, that is the monoglycerides and the free fatty acids. And after getting in, so these monoglycerides and free fatty acids are resynthesized into into triglycerides inside the epithelial cells, and these triglycerides aggregate and are coated with a layer of lipoprotein from the endoplasmic reticulum. Here you can see, to form the water soluble chylomicrons. And these chylomicrons gets out of the enterocyte by means of exocytosis, so which is water soluble, and these chylomicrons are unable to cross the capillary membranes, so instead, so it is not easy to reach, so instead it reaches the central lacteal, which is very nearby, which is in the center, and it gets into the central lacteal, and which moves to the thoracic lymphatic duct, and then the portal vein. From the portal vein, it goes to the liver for further absorption. Coming to the fluid and electrolyte absorption and secretion. So, fluid entering and exiting the gut. So, the volume entering is the diet we take around 2 liters. So, saliva is 1 liter from the stomach, gastric secretions 2. So, bile is 1 liter, pancreatic secretions are 1 liter, and from the small intestine it gives around 2 liters. And again, the volume absorbed or so from the duodenum and jejunum around 4 liters, and ileum 3.5 liters, and from the colon it is 1.4 liters, and only 100 to 200 ml of fluid is excreted every day. Mechanism of water absorption. So passively and isoosmotically following osmotic gradient due to absorption of electrolyte and nutrient. So the absorption of water happens. So in duodenum it happens due to the chyme. When the chyme enters, so the osmotic gradients pull more fluid, and in jejunum and the ileum, the reabsorption of the sodium chloride, which takes away the fluid further from the intestine. So water movement in the small intestine, water moves in and out of the gut lumen by diffusion in accordance with the osmotic process. So if the chyme is hypotonic, the water is absorbed. If the chyme is hypertonic, the water enters the intestine. So the fluid is secreted into the intestine. So it depends on the chyme how we live. So usually the chyme is isotonic. Mechanism of water absorption. In small intestine, there is sodium glucose co-transport, sodium amino acid co-transport, and sodium hydrogen counter-transport. In colon, passive diffusion happens through the sodium channels, which is stimulated by the aldosterone. So aldosterone acts here also. Sodium absorption causes the water absorption. Sodium is absorbed by the epithelial cells of the small intestine. Sodium uptake creates negative electrical potential in gut lumen that provides gradient for the chloride uptake. So sodium uptake happens, then along with the sodium, chloride uptake happens, and water follows the sodium chloride. So that is in accordance with the osmotic process. So sodium absorption in the small intestine. Sodium is absorbed by 4 mechanisms: diffusion, taking the diffusion through the water-filled channels, and co-transport with the amino acids and glucose, co-transport with the chloride, and counter-transport with the hydrogen ions. So aldosterone increases the sodium reabsorption and potassium secretion, in the small intestine in the colon. Absorption of vitamins. So in terms of absorption, vitamins are classified to whether they are lipid soluble or water soluble. So the fat soluble vitamins are A, D, E, K, and water soluble vitamins are B complexes and vitamin C and folic acid. Thiamine is absorbed by sodium dependent active process, but in high concentration it can happen by passive process also. Vitamin C gets absorbed both by passive and sodium dependent active transport. Folic acid is transported actively in duodenum and jejunum. Cobalamin absorption, it requires specific factor which is called as intrinsic factor, which is secreted in the stomach. So the absorption of vitamin B12 is unique among all the vitamins. So the fat soluble vitamins are incorporated into the vessels and absorbed along with other lipids. Absorption of fat soluble vitamin depends upon the absorption of other lipids. Vitamin A gets ingested as beta carotene and gets absorbed as such, and vitamin D and E are digested by cholesterol ester hydrolase, and vitamin K1 absorbed by active transport mechanism. Vitamin K2 is taken by the passive transport. Fat soluble vitamins appears in the exocytosed chylomicrons. Absorption of vitamin B12, which is very unique. You see the dietary vitamin B12, it enters the stomach. Here you can see the intrinsic factor, and there is also a R binding protein which is called as haptocorin. So there are two proteins released. One is the R binder or the haptocorin, and second one is the intrinsic factor. So among these two, the R binder has higher affinity to bind with the vitamin B12 or the cobalamin. So once it enters the intestine, so the pancreatic enzymes, they cleave the R binder. Then after the cleavage of the protein, after the cleavage of the R binder, so by the pancreatic proteases, the intrinsic factor binds with the vitamin B12. Then when it reaches the brush border of the intestine, so this intrinsic factor has specific receptor in the brush border. So without the intrinsic factor, the vitamin B12 alone will never get absorbed. It can get absorbed only along with the intrinsic factor. So along with the intrinsic factor, this complex enters the enterocyte, and finally, so it binds with the transcobalamin 2, and it is a binding protein and gets into the blood vessels. And there is another binding protein called as transcobalamin 1. So while getting into the liver to get stored in it, it binds with the transcobalamin 1. So in the blood, it binds with the transcobalamin 2, and in the liver to get stored, it binds with the transcobalamin 1. Absorption of iron. There is heme iron and non-heme iron. So heme iron is from the hemoglobin and the myoglobin, whereas the non-heme iron is from our diet source. So heme iron, which gets into the enterocyte with the help of heme transporter, whereas non-heme ion which we take is in ferric form, but the most favorable absorbable ion form is the ferrous form. So this ferric form gets converted into ferrous form with the help of reductase, and with the help of ion transporter, this ferrous form gets absorbed, gets into the enterocyte. Then the heme which gets into the enterocytes gets converted into the ferrous form of ion with the help of heme oxygenase. And the heme by means of active transport gets into the capillaries, and the ferrous form of the ion along with the apotransferrin forms the transferrin and moves to all the tissue through the blood. So wherever the requirement is, it goes to that tissue. And the remaining ferrous form gets converted into ferric form, and along with the apoferritin it forms the ferritin. Some of the ferritin sheds off and some stays in the enterocyte until the enterocytes sheds off. So the malabsorption syndrome, multiple nutritional deficiency states are produced. So features include general weakness, signs of hypovitaminosis, iron deficiency anemia, steatorrhea, so undigested fat gets excreted in dehydration. Conditions causing malabsorption syndrome, celiac disease due to the deficiency of gluten hydrolase, so gluten intolerance in tropical screw, so that is a malabsorption of vitamin B12 and folic acid. Crohn's disease is inflammatory bowel disease. Resection of small intestine affects the absorption of the nutrients. So gastrocolic fistula, so that also in that the food bypasses the small intestine, so the malabsorption happens. In blind loop syndrome, so the absorption greatly affects here. So the formation of areas of intestine where bacteria can proliferate without being subjected to move down the intestine. So there will be pouches formed by the bacteria, so absorption may not happen. So to summarize, so from the saliva, the digestion starts with the salivary amylase, so which digests the polysaccharides and disaccharides into the oligosaccharides, and in the stomach only the protein digestion happens with the help of pepsin. In the pancreas it secretes the pancreatic amylases and the pancreatic trypsin, chymotrypsin and carboxypeptidases, so which digests the larger protein molecules into small peptides and amino acids. And then the nucleic acid digestion also happens in the small intestine with the pancreatic nucleases into nucleotides, and the fat digestion also happens in the small intestine with the help of the pancreatic lipases. And in the small intestine, in the brush borders, there is disaccharidases, so which forms the monosaccharides, and it also the small intestine also has the dipeptidases, carboxypeptidase and aminopeptidases to act on the proteins, and it also has the nucleidases finally to give the pyridines and purines. And the fat digestion, so the bile salt is very important for the fat to get digested, and villi plays an important role for the absorption of the fat, fat molecules, and finally it gets into the lymph in the form of chylomicrons. Books to read: Guyton and Hall textbook of medical physiology, Lennon's Review of Medical Physiology, comprehensive textbook of medical physiology by Professor G. K. Thank you.